In 1965, the first adeno-associated virus was discovered as a contaminant of adenovirus preparation. Adeno-associated virus is a single-strand DNA virus, belongs to genus Dipendo-parvovirus. Why adeno-associated virus is widely used in gene therapy? This is because this virus is non-pathogenic and they are able to specifically integrate into the host cell genome at a specific site in the human chromosome 19. Besides that, this virus is also able to infect dividing and non-dividing cells of human and some primate species. There are two modes of adeno-associated virus life cycle, which is either the lysogenic or lytic cycle. The adeno-associated virus infection begins with virus binding to cellular receptor heparin sulfate proteoglycan. Internalization of the virus is enhanced by interactions with one or more co-receptors, for example, integrins and fibroblast growth factor receptor 1. After the adeno-associated virus successfully entered into the cell, the viral genome is being transferred into the nucleus. They use the host cell machinery to form double-stranded DNA and then integrate into a locus specific localized on the long arm of human chromosome 19. When helper virus is absent, the integrated viral genome as a provirus remain in a latent form. This is what we call the lysogenic life cycle. While when the helper virus such as herpes virus or adenovirus is present, the adeno-associated lytic phase begins with the transcription of viral genes, followed by viral genome replication, and lastly, packaged into capsids to produce new viruses. Apart from that, in order to use adeno-associated virus as a vector in gene therapy, recombinant adeno-associated virus must be produced. There are two ways can be used to synthesize recombinant adeno-associated virus. The first method is by using the helper-required method, which is the regular cell cycle of adeno-associated virus that I had explained just now. Another method to produce recombinant adeno-associated virus is transient transfection of all elements that are required for recombinant adeno-associated virus production in host cells. This method required three elements. First is the adeno-associated virus inverted terminal repeats containing plasmid carrying the gene of interest. Second, the adeno-associated virus wrap and cap containing plasmid, and plasmid containing isolated helper gene from adenovirus. Both of the methods have their own advantage and disadvantage. In terms of helper required method, this method can be easily scaled up in culture so that large amount of recombinant adeno-associated virus can be produced. However, contamination with the wild-type adenovirus can be happen, which can cause safety problems. For helper-free method, of course high concentration of recombinant adeno-associated virus that are free of adenovirus can be synthesized, but the process to produce this recombinant virus is very labor-intensive and expensive.